Hello everyone. In this lecture, I will talk about the evidence-based practice. Sometimes I will use a short term, EBP, to represent this concept. This is the agenda of this lecture. I will first talk about what evidence-based practice is. Then I want to show everyone the barriers to EBP and the resources that can facilitate EBP. Next, I want to talk about what evidence is, and then what techniques we can use to collect evidence. In this lecture, I want to focus on one technique called meta-analysis. Let's talk about what evidence-based practice is first. The development history of EBP can be dated back to 1970s. At that time, Dr. Archie Cochrane developed a new concept in the medical field. According to Dr. Cochrane's belief, not all medical or clinical interventions are needed to cure a disease. In his opinion, some medical or clinical interventions may do more harm than good to patients. Therefore, before applying some interventions to patients, the healthcare professionals should evaluate the validity and the effectiveness of some interventions in order to get the best solutions to the patient's medical issues and provide best quality healthcare to the patients. Dr. Cochrane's idea may sound a little far away from us, but if you think about it, we are getting benefits from his idea almost on a daily basis. If you are on some medication, when the doctor prescribes the medication for you, he or she will also tell you the amount of medication you need to take every time. Let's say 10 milligram. Have you wondered why is 10 milligram not 5 milligram or 20 milligram? The reason is, before the pharmaceutical company starts selling the new drug, the company has done some trials among a small group of patients, let's say 50 or 100 patients. The company will give different amounts of medications to the patients and monitor their responses when they take different amounts. According to some statistical analysis, the company can eventually make a conclusion that 10 milligram works the best in this sample. You remember we studied the concept of the level of significance. We use this concept to infer the findings from sample to the population, right? In medical trials, we use the same concept. If we find that 10 milligram is a significant amount, we can predict that any patient in the population will have a similar effect. So if the pharmaceutical company finds that 10 milligram works the best in the trials, and it is a significant amount, the company will ask every patient to take 10 milligram every time. Not more, not less, in order for every patient to get the best results from the medications. This is the example of Dr. Cochrane's idea. This is also an example of the evidence-based practice. Let's give a formal definition to EBP. EBP is a practice that focuses on the conscientious, explicit, and judicious use of best evidence in a healthcare professional's decision-making, in order for him or her to provide the best quality healthcare to patients. Several new subjects have been established based on the evidence-based practice. For example, clinical informatics. Similar to nursing informatics, clinical informatics is also a combination of information science, computer science, and medical knowledge. The clinical informatics professionals will usually collect evidence by comparing the performances and responses among different groups when they are given different medical treatments. After collecting the evidence, the clinical informatics professionals will use the statistical analysis models to compare the results from different treatments. Then they will make recommendations about the best solution to the physicians or pharmacists. Another subject is called Translational Bioinformatics. This subject takes one step further. It focuses on finding the best medical solutions by collecting and analyzing biomedical or genomical evidence. In cancer research, a new treatment called targeted therapy is getting popular. Some pharmaceutical companies have developed new medications that intend to grow certain proteins in a patient's body. Those proteins can stop the growth of cancer cells. How do we know those new medications are effective? What we can do is to compare the patients who took the medication and the patients who didn't. Then we can make a conclusion if this new medication, the new targeted therapy, is effective. This is an example of translational bioinformatics. The evidence-based practice sounds like an amazing idea, but it is not accepted by every healthcare professional in practice. EBP may face challenges or barriers in practice. For example, a healthcare professional may lack of knowledge or motivations of adopting EBP. A healthcare organization may not have enough financial resources to collect and analyze evidence all the time. Physicians or patients may have a resistance to EBP. A healthcare professional may only have a limited access to the up-to-date and user-friendly technology or computer system for analyzing and collecting evidence. A new department called the Agency for Healthcare Research and Quality, or ARC, has been funded and developed by the US government in order to encourage the adoption of evidence-based practice. ARC provides several useful resources. For example, the National Guideline Clearinghouse, or NGC. NGC is a comprehensive database that shares guidelines and documents for adopting EBP with practitioners. ARC is responsible for updating the documents and guidelines. You can use the web address on this slide to subscribe to the NGC email list in order to receive the latest updates from this website. In addition to government resources, there are some free public resources that support EBP. For example, PubMed for HH. This is a free mobile app. Smartphone users can use it to search the latest studies and research in the database of the National Library of Medicine. For example, if you are interested in targeted therapy, once you type in the keyword targeted therapy, the mobile app will retrieve the abstract of the latest study in this field on your smartphone. Obviously, collecting and analyzing evidence is the most important topic in an evidence-based practice. So what is evidence? Simply speaking, evidence is the data that we can observe, collect, and analyze. The next question is, how can we collect evidence? According to Dr. Cochrane's idea, using the randomized controlled experiment is the best technique. Notice that the keyword here is randomized. Randomness is very important to a successful experiment. Randomness means that when you organize an experiment, you cannot recruit patients from only one demographic group. You must recruit patients from a variety of demographic groups. Why is that? Because if you only focus on one demographic group, the findings from your experiment is narrowed. You cannot apply your findings to other groups. For example, if you want to test the effectiveness of a new medication, but you only focus on patients with high incomes, you will get a problem. The problem is, the patients with high incomes may be able to afford additional supplements. Those supplements may work better with the new medication. Then when you apply the findings from this group to patients with low incomes, they may not be able to afford the additional supplements. You cannot apply the findings to those groups. That's why it's very important to recruit participants from different demographic groups in order to eliminate the additional influences in our experiment. The randomized controlled experiment is the best technique, but it may not be always available. For example, because of the privacy concerns, you can only recruit patients that are willing to participate in your experiment. If the randomized experiment is not available, we have to develop some other techniques to perform evidence collection and analysis. The alternative technique is called meta-analysis. It is frequently used in practice, so let's talk about this technique in this lecture. Simply speaking, a meta-analysis is a comparative study of previous results. When you use meta-analysis, you usually will go through the following steps. First, identify the problems or questions you want to answer. Let's say you want to test the effectiveness of a medication. This is the question you want to answer in your study. Next, instead of organizing an experiment on your own, you want to look for the previous studies that have been done to answer the same question. Before us, other researchers may have already organized the experiments and tested the effectiveness of the medication. We want to search the databases on the NIH website or the National Library of Medicine website to look for the previous studies. Once we get the previous studies, we want to summarize the findings from each study. Next, we want to compare the findings from different studies and make a conclusion that when the medication works the best according to different conditions. This is the process of meta-analysis. Meta-analysis can be useful, but it could face some challenges as well. For example, we are not organizing our own experiment, so our findings from meta-analysis can only be as good as previous studies. This is a limitation of meta-analysis. That's why meta-analysis is only used as an alternative when the randomized controlled experiment is not immediately available.